Hi everyone, my name is Aisha Nusra Sanjar, as you can see here, and today we're going to talk about DNA profiling utilizing the PCR of STRs and how it is useful in criminal cases. First, I'd like to start by defining the STR. These are short for short tandem repeats. Short tandem and repeats, the name implies everything it needs. These are short sequences of DNA. They can be from 1 to 13 nucleotides. And these are repeated tandemly, like this STR made up of CA nucleotides and 1, 2, 3, 4, 5. This is the number of repeats for this certain STR. And these are found in DNA, and in every person they can be of different numbers, and this variance in numbers are caused by the errors during replication. They can be multiplied, they can be deleted, because as the DNA is lined up, it is hard to place it like in here, for example, CA, the DNA strands can be aligned like this or like this, etc., and they get increased or decreased. These STRs are very useful in criminal cases because, as I said before, each person has a different set of numbers of STRs. And to define the numbers, we do PCR first from the DNA, and then we run it on electrophoresis. During the PCR process, the primers are used that are aligned with the flanking regions of these STRs. And if you know how to do PCR, it's a really quick and easy thing. As you can see in this PCR result that is run through a gel. On the second lane, we can see a single STR, and on the third, we can see again a different STR. These are from two different people, and we can see that they have different STRs, but they share one line. And for the fourth lane, we see a multiplex PCR. If the STRs you're interested in have different lengths, you can use them all in the same lane, and you can have three distinct STR places on the gel. And then to do the analysis, you don't have to run it through a gel electrophoresis. You can do a capillary gel electrophoresis, which results in this picture actually. Like in A, you see it is the biggest one, and it is mostly the last one that passes through the capillary. And this picture actually is known as an electropherogram. If you look at each of the STR of a certain person, as we see in this picture here, this is the full DNA profile of the STRs we are interested in a person. You see the peaks in here. These are all the STRs we're looking for. And the last part is about if this person is male or female. Now, we have covered how to amplify these STRs and then run them through the gel and then generate a profile. So how does it work in criminal cases? As we said before, we collect the DNA. These collection of DNAs can be of anything like blood, saliva, or hair, or just scraps of skin. And then we extract the DNA, as we learned in previous years. The PCR amplification is done, the profiling is done, and then we put the profile on databases. In United States, the CODIS, which is the acronym for Combined DNA Index System, is used for this. you upload your profile in here, and then hopefully you get a match, and then you follow that lead, and then if you find a suspect, you need to test to confirm that STR is from that suspect. And to do that, for example, let's take this example from forensicscienceamplified.org. Here you have a mixture. There's a lot of DNA in it. And then you have the suspect. You see the suspects, STR peaks like this. And in the mixture, there are a lot of them. But you see that these match with the suspect. And when you see this, you just get the suspects, the toothpicks. You get something from their mouth, and then you just have these peaks on your suspect. And then how it is useful, like it is uploaded in this database, and it stays there. And even if you found the evidence and you cannot find a match, there is a possibility that in the future the match can come. And from an article written by Stanley and Associates, there's a case report known as Case Report 1. This is from Kichevo. I guess it was a city in Macedonia, right? Yes. A couple was found killed, and the murderer roped their bodies, like the bodies were tied. And in the autopsy, they have extracted different DNAs from both the victims, the rope that was used, etc. But when they uploaded their profile to the forensic database, they couldn't find any matches. And after a few years, 5 years to be exact, there was a charge that was burglared, and then during crime scene investigation, a suspected person's blood was found on the window. And this person was arrested, and then there was the buccal swab, like they took a toothpick in his mouth. And then the DNA of that suspect was recorded in the database. And resulting from that, this married couple that was found dead was also solved, because this person, this exact person that has done the burglary, was the one that was found that had the DNA of these rope and glow. If you take a look at this table, this is also in, these are all STR locuses. You see victim 1 and victim 2, and then there are rope and glows. These numbers represent the STR peaks. And if you take a look at these section, like the rope glows versus the suspect, they exactly match at all STR positions. So the suspect was caught. So that's pretty much it. It's a really cool thing to work with, in my opinion, because it is so easy. Like, there are some nucleotides that are just repeated on, and then that makes it your fingerprint, just like this, a molecular fingerprint. Thank you for listening.